I'm talking today about developments in metabolomics. So these are the four areas that I'm going to be covering. I'm going to talk about just the description of metabolomics because there may be several. So what am I referring to? Metabolomics and dietary intake. How do you use it to identify what people are eating? The impact of nutritional deficiencies or excess on metabolomics. How do they impact it? And metabolomics and nutrient patterns related to disease. Can you use metabolomic assessment of nutrients to identify risk? So this first slide defining what I mean by metabolomics. I'm pulling this from an article that Dr. David Brady and I wrote last year on applied metabolomics. So the term metabolome is about 20 years old. It's really metabolomics is the scientific study of chemical processes involving metabolites. And the metabolome is that complete set, just like the genes and the genomes and proteins and proteome. So a metabolic signature is the measure of metabolites describing an individual's current status, which certainly can change. So additionally, I've seen some research and some researchers noting that getting a metabolomics review every year should probably start to become part of a yearly physical. So you can act as your own baseline and know what's right for you. So I'm putting this study up because it worked with inborn errors of metabolism, so IEMs. So inborn errors of metabolisms uses older technologies, and they used to look at, are there inborn errors of metabolism, generally with infants. And they were primarily looking to see if a marker was there. So the enzyme was just gone. So you're thinking PKU or maple syrup urine disease. So the enzyme is missing and the marker's super high. That's all they were looking for. And then functional medicine came along and they were like, hey, why don't we look and see if there's a normal bell curve? Like what's a normal person have with this metabolite? So, when they were started doing inborn error testing, they wouldn't even have thought of looking for a normal level. So then functional medicine came in and they made hypotheses and assumptions based on pathways and some primary research. And they developed stories on nutrient needs and nutritional tests. And then metabolomics has come along and everyone started looking at a bigger picture to step back and identify a full range of pathways. And so there's a lot more research now. And then getting back to inborn errors of metabolisms, this study was done in 2019, and they found that when they used traditional metabolic screening assessment, they had a pretty low 1.3 identification. But when they used diagnostics with metabolomics, it increased sixfold, and they found some new ones. So even there, it can work. So when I think of organic acid testing, I always lovingly think of the Krebs cycle. So the Krebs cycle and the amino acids that flow into it. But when I think of metabolomics, I think of digging a little deeper. So I'm going to use tyrosine, phenylalanine tyrosine into the Krebs cycle at fumarate or fumaric acid. And so it just throws a wider net. So from that organic acid, from the Krebs cycle, I'm going to go here. So I'm looking at this full picture. So phenylalanine with phenylalanine hydroxylase and BH4 goes over to tyrosine. That can, with its tyrosine hydroxylase, it can go down with neurotransmitters, or it has a lot of other pathways. And one of them is going to 4-hydroxyphenylpyruvic acid, to homogenistic acid, into the Krebs cycle. So that's how it gets in there. Homogenistic acid is one of those markers that if this enzyme is missing, this builds up and you can get diagnosed with alkaptonuria. But even research on this marker has identified that it has a normal bell curve. So a lot of other things that you can look for. Phenylalanine, if it can't go over to tyrosine, will build up and can break down to phenylacetic acid. And so sometimes people take supplementation with phenylalanine and they get dizzy or something. It may be that they have an inability to break it down over here in this pathway. It's good to look at cortisol because norepinephrine to epinephrine is impacted by cortisol. It's also good to look at the breakdown with gut microbiome. So p-hydroxyphenylacetate It gets decarboxylated to P-cresol. So all that's to say, when I'm looking at metabolomic stuff, I want to see a full pathway. I want to see, phenylalanine and where it is. So this phenylalanine, it's kind of going over to tyrosine, and I don't know if that tyrosine is getting used up over with the homovanillic acid, which is a dopamine breakdown marker, DOPA, or if it's just not going over all the way. So I can look at all these markers. I can look at the 4-hydroxyphenylpyruvic and the homogenistic. I can look at fumaric acid, the impact of cortisol, and the 4-hydroxyphenylacetic acid, identifying bacterial fermentation. So if it were really high, it would be staying more in the gut. So all that is so moving from nutritional metabolomics to nutrametabolomics. So this is a good article. It's looking at metabolomics, over this 20-year period, that it is an emerging field. And so we're moving from major and micronutrient kind of sources of identification of need to determination of health risks. And you see that with a lot of these. So this is looking over the last 10 years. There's been a big increase in the type of studies people are doing. And, while it might have been started out with just blood and urine, over the years it's increased. So obviously more stool testing, saliva, and others. Looking at dietary assessment, like how do these correlate with dietary assessment or gut microbiota? Looking at different nutrients and food groups and dietary patterns. There's a lot that they look at. So, individual proteins, amino acids, carbohydrates, and these have gone up. Right now, there's a lot of work on fats and fatty acids. There's still a lot going on with proteins, carbohydrates, and then individual foods, dairy foods, legumes, cereals, nuts. So looking at them all. Some of them might have to do with who's got the biggest lobbying group. And then multiple food groups, because there's a lot of synergistic effects. I think of, plantains or black walnuts can both increase 5-hydroxytryptophan or 5-HIAA, the breakdown product. And when you have them together, it's a synergistic effect. So looking at all those things. And then looking at what about different diet types, Mediterranean, vegan, low carb, gluten-free, all these things and how they have an impact. Certainly gluten-free is known to decrease bifido content. So looking at all these and their relationship. And then kind of the big where to go is what about with disease? So you can see there's a lot of looking at metabolic syndrome, looking at hyperlipidemia, cardiovascular disease, obesity, those things that we're generally getting a lot of. So during this talk, I'm going to talk about, I have a few slides that have some markers, and it's going to be with a case that I had. So it's a 28-year-old female. She works as an accountant. She has some bloating after meals, some anxiety and attention issues. She exercises most days. She, says she eats a pretty good whole food diet, plant-based milk with some cheese and meat. So what I like to do when I see somebody who wants a whole comprehensive assessment, I look at organic metabolomics, which you can order from Diagnostic Solutions. I look at GI map to look at the gut, what's happening there functioning, and what are the microbes, and then a genomic insight, because it gives me a full picture. So when you look at her, she's relatively healthy. She's going to work. She kind of feels okay. So just her stress seems to be, so when she says anxiety, that seems to be standing out a little bit. Her metabolic processing, nutrition, microbes, metabolism looks good, but she's got some things going on with the amino acids. So I'm looking at these 6 different patterns. So this is answering my second question. So what are people eating? So using metabolomics for the simplest things as markers of what people are eating. So this is probably going to be familiar to a lot of people. So urine fructose levels increase with fructose intake. And so looking at how much fructose is somebody getting in their diet, which she's getting 11. So I have this that I have made. I use BioCare and then I pull this out. So her fructose intake. And so her intake, which falls about the, almost the 25th, and her urine level are kind of matched there. So I can see, I get a better picture. So she's not a big fructose eater. The problem with fructose is that unlike glucose, fructose is absorbed in the intestine independent of energy or sodium exchange. So when it's consumed in high amounts, and it doesn't take long, a Coke or something is going to send it over the edge. Fructose gets transported to the liver and then is preferentially converted to lipids. So hyperglycemia has been associated with increased fructose levels, especially in patients with diabetes. So some other things I look at. So there's several of these studies that look at all the different foods that can be in these levels. So foods that may be related to hydroxyproline or glutamic acid, the leucine, isoleucine, and valine, the branch chain amino acids. They're more likely to be in milk and red meat products. Beer was associated, so maybe people are eating these together. So there's several studies out there looking at these associations. But when I look at them, I want to look at them together. I want to look at groups because you can't just have one that comes in and not impact the other. So methionine produces cystathionine, cysteine produces taurine. Cysteine is typically present in its oxidized form, cysteine, in extracellular spaces, and that's got to have to get in, it's got to have a glutamic acid transporter. So it's important to look at the whole picture. So foods high in cysteine can impact all of these. Apologies. So looking at the cystathionine, that's a little bit higher. The methionine is a little bit higher, and kind of looking at those and what impact diet can have. So looking back at her diet record. This is something, if you love the Human Metabolome Database, HMDB, you're going to love the food database. So you can look up any metabolite and then what foods may increase it. So that's just a good kind of find. So beyond specific foods or individual, and then groups of metabolites, research has been associated with different patterns of disease with types of diet. So metabolomics is trying to just look at the whole system. So you can see this one. They look at a low meat versus a high meat. They look at people who are high fruit intake. So there's all different combinations, and they're looking at what pattern you're going to see. I think this is important to have this, and then along with diet intake. There was a recent study looking at cheese and cardiovascular disease, and they used a marker they said was for cheese intake, but some argue that it might actually be for fiber intake. So it's good to have a diet assessment. So these are some of the things that are coming out. So some of the markers that you'll see with a vegetarian diet. So citric acid is going to be high in a vegetarian type diet. And then p-hydroxyphenylacetate, so the breakdown with gut microbiome. A ketogenic diet is going to have some of these markers that are different. And then the Mediterranean diet, which has quite a bit of research. So, it's not a surprise that a ketogenic diet would have high beta-hydroxybutyrate because that is, ketone markers. But there are some newer ketogenic diets that are vegan and high in fiber. And so their picture would be a little bit different. So here's a study that was done in nutrients. And they looked at Mediterranean diet and the impact it would have on metabolites and associated. So increased tryptophan, increased hippurate, p-cresol, and a decrease in branch chain amino acids. And then they also looked at the impacts that a Mediterranean diet would have on microbiome. So Prevotella is known to increase with a plant-based diet. They also found an increase in bifido, in Clostridium marker, interestingly in Candida, and Faecalibacterium, and a decrease in Lactobacillus and Firmicutes. So those are interesting kind of finds. So this study actually went further, and they looked at diet and used metabolomics and genetics to assess metabolic signature. It's the first to develop a metabolic signature for the Mediterranean diet based on metabolomics profile. So they are demonstrating that the level of dietary adherence along with an individual's response to diet can be objectively measured. So they identified that it was adherence to that could predict future cardiovascular disease risk independent of traditional risk factors. And they did Spanish and US cohorts. So they note that they had pretty good reproducibility of the findings in both US and Spanish population. And it does their robustness. So they looked at the diet. They actually looked at something, the Mediterranean diet inherit screener. And then they looked at genetics and they looked at a metabolic signature. So some of these research are looking at how you tell the difference between some of these. So they looked at the alternative healthy eating index, the dietary approaches to stop hypertension, and the Mediterranean style diet. With the proteins, Mediterranean diet did not really correlate as much as the alternative and the DASH. But then when you looked at the metabolites, they all kind of had similar. So just kind of parsing out those type of things is what you're seeing. So next, my other question was, how do nutrient deficiencies or excess impact metabolomics? And metabolomics is, trying to shift away to avoid disease, to getting the right nutrients and optimize overall health and biochemical function. So what pattern do you want to see? So this is probably something common to people. They're used to looking at, a lot of clinicians have probably looked at fig glue for folate deficiency, and if that's a thing. So looking at folate on its own can be helpful, but urinary histidine excretion, maybe you're adding blood histidine, may be a way to help increase that. So in this study, they looked at folate deficiency, B12 deficiency, and normal controls. They look at baseline histidine, baseline fig glue, and then with the histidine load. And they did find that excretion of histidine, this is urine histidine, was like 3 times higher in a folate deficiency. And a B12 deficiency was a little bit higher. So you're going to see urine histidine go up, but you would see plasma histidine go down. So interestingly, just when someone is folate deficient and you give them histidine, it can take away their anemia because hemoglobin is about 10% histidine. So adding it can be helpful. Another thing that people are probably used to are using tryptophan markers, xanthurenic acid, and kynurenic acid to look at need for B6 because the KAT enzymes both require B6. So that can work, but really they're learning much more about this pathway. And it's beneficial to have the whole pathway to kind of see what's going on. So here is this is a compilation of a couple of studies. Some of them are metabolomic load, like tryptophan load, and some of them were mathematical modeling. So this marker here is looking at, just a steady state, a plain B6 deficiency, and then oral contraceptives with the load. So you'll see that, as mentioned in this study here, if you get a moderate deficiency, hydroxykynurenine, which is HK, will increase. It's also 3-HK is the same as that. And kynurenic acid and anthrylic acid will decrease. So if you have more severe deficiencies, you'll get an increase in kynurenine and xanthurenic acid. You'll also get increases in glycine, serine, and cystathionine. So looking at xanthurenic acid may not be enough. Also, it's important to look at microbial metabolites, which can let you know what people may be eating and how their gut bacteria are responding. So you eat some soy, you're going to end up with phytoestrogens like equal. You eat fruits and vegetables, whole grains, you're going to end up with these phenolic metabolites. You get amino acids, protein, you're going to get these amino acid compounds, so bacterial action on these. So this also is a great article. So looking at polyphenols are excreted in the urine, their interaction it's a two-way interaction with the gut microbiome. So polyphenols are going to support the growth of microbiota, and then the microbiota act at the polyphenols to increase their bioavailability and produce compounds like hippuric acid or equal or 4-hydroxybenzoic acid. So just looking at this one, so 4-hydroxybenzoic acid is the result of bacterial fermentation of anthanocyanins by generally Clostridium or Eubacterium. Sometimes the research is found just total microbiota. But they're looking at, you know, those anthocyanins. I'm just going to say purple, blue, and blue-greenish type foods. So those are they're going to act on those. So foods like huckleberry, coriander, blueberry. And then, so this is from our accountant, and she's a big vegetable eater. So it's not a surprise that she has these a little bit more elevated. Also, 4-hydroxybenzoic acid was one of 3 urinary metabolites, along with histamine and azelaic acid, that was able to discriminate between a low and high FODMAP diet in IBS patients. So p-hydroxybenzoic acid increased on a low FODMAP diet. So isoflavones. So equal is a great marker to identify how isoflavones are being metabolized. The ability to produce equal varies among individuals because only people who have the right intestinal bacteria, and I think it's a group of about 8 of them, are capable of producing equal and are regarded as equal producers. So if you grew up in a population that ate a lot of soy and tofu, then your bacteria would develop that way. And you would be more likely to be an equal producer. Vegetarians are going to be more likely to be equal producer than non. I've seen one study where they tried to convert people, and they did some. Spot urine equal levels have been found to correlate strongly with serum concentration. And this is identifying that high is not a problem. It's just identifying that statistically, this is higher than we normally see it, but it's not an issue. I mean, I wish I was an equal producer. Isoflavones are still beneficial even if you don't produce equal, but you don't want to assume you're getting equal if you don't. So nutrient deficiencies can result in oxidative stress and inflammation. So the 2 of the big ones I think of are 8-hydroxy-2-deoxyguanosine and the kynurenine to tryptophan ratio. So I always think of that with nutrient deficiencies. So they can lead to increased oxidative stress, which itself can cause issues. So this goes to my third question. It's my third one. How can metabolic assessments of nutrients identify disease risk? So this is really what people are looking for. So there's quite a lot of information on this. There's significant research. There was a study in 2020. They did a meta-analysis of 46 articles, and they found that the analysis indicated that people who had higher branched chain amino acids and aromatic amino acids had higher risk of type 2 diabetes. There's also a 216 study with metabolomics in prediabetes and diabetes, and they did a systematic review and meta-analysis. So these are both big studies, and they found the risk of type 2 diabetes was higher if you had higher branched chain amino acids and tyrosine and phenylalanine. Also alanine tends to. So if you think of pyruvate comes into the Krebs cycle, but it can also go to alanine or to lactate. So alanine and lactic acid have also been associated. And lower levels of glycine and glutamine may be protective. And another review in the Women's Health Study found that plasma branched chain amino acids were positively associated with cardiovascular disease in women. And that's a pretty big study. They had like 27,000 people. So there's a lot of, elevated blood concentration of the branched chain amino acids is an established biomarker and prognostic factor for obesity, insulin resistance, and type 2 diabetes. The Framingham Heart Study found that fasting amino acid levels of these branched chains were elevated up to 12 years prior to diabetes. So they may be identifying kind of a low level metabolic impairment. And the future risk of diabetes was elevated at least fourfold in those with the highest plasma amino acids. A single fasting measurement of these amino acids provided information incrementally over standard risk factors. So these are statements from this study, Dr. Wang, and it's an interesting article because she then tries to get people to decrease branched chain amino acids by 50% just using diet. So she had them restrict branched chain amino acids by 75% within the diet by just lowering overall protein intake. So they selected a lot of foods that are low in branched chain but had protein, so a lot of vegetables. Secondly, they used foods that are just naturally low in proteins, and then they got some commercially available low or no protein foods, the stuff that somebody with an inborn error might use. And this is kind of what they found. So this is the research study. And then this is a 7-day low branched chain amino acid diet. And it came down, and each of the amino acids came down. So I'm comparing this, like Mayo has a range, LabCorp has a range, and this is Journal of Epidemiology. And these single numbers are just the mean. So then the Organics Metabolomics from Diagnostic Solutions also have a very similar range. Most places are. So we've included total branched chain amino acids so you can kind of see where people are and you can compare it to research studies. And then this is our accountant again, and her level of branched chain amino acids were lower. The study was only 7 days, so they didn't find a significant, they did lower insulin resistance. So then these are just some more studies that continue. This is one that came out in Biomolecules in 21. So just looking at the kind of advantages, disadvantages of 2-hydroxy butyric acid or butyrate. So it's associated with insulin resistance. And then again, the aromatic amino acids. So hers are, not sure why that phenylalanine is a little bit elevated, but overall, they're all within range. They look at the branched chain amino acids and then glycine because it's low, it can be a problem. So associated with increased risk of metabolic disease, and hers is pretty low. So then we're going to ask, some questions of why. It doesn't appear to be solely due to diet. So that branched chain amino acids become elevated. Some of it's due to metabolic processing. So leucine is a branched chain amino acid and has specific metabolism related function. So the mechanistic target of rapamycin complex, or the mammalian target of rapamycin, its name changes, is an essential regulator of cell growth and metabolism, and it's regulated by several environmental cues, including amino acid availability. So also growth factors, energy levels, stress, and it's all done to, coordinate cellular status with environmental conditions. So if there's a dysregulation of mTOR activity, of mTOR is closely associated with various diseases. Besides diabetes, there's been research on cancer and neurodegenerative diseases. So high blood levels can cause that. So when you look here, a high leucine, high glycemic, really Western diet can increase leucine. Things like metformin, the drug that it can antagonize leucine so it can stop its activation, so it keeps it inactive, can cause that. It can also lower glucose. But there are also other things like resveratrol, EGCG, DIM, things that can also decrease its activation. Plus, bariatric surgery in obesity is known to reduce plasma levels of leucine and other branched chain amino acids. And interestingly, when you look at it, the baseline levels of branched chain amino acids are now being used to assess who may benefit from a diet. So in patients who began a Mediterranean diet with a low baseline branched chain amino acid had higher probability of type 2 diabetes remission than those with a high baseline branched chain amino acid. They didn't find an association with a low-fat diet. So the study is suggesting that measuring branched chain amino acids can potentially be used to select who's going to be the most responsive to diet. And I wonder if some people are kind of through lifestyle are causing their branched chain amino acids to be elevated, and you can fix that. And some people just metabolically have different functions going on, and diet may not be as big an impact. So another area that that is kind of hot out there is looking at tryptophan and looking at the kynurenine to tryptophan ratio. So tryptophan goes to kynurenine, and it also, besides that, it can go to serotonin, or it can get broken down and metabolized by gut bacteria. So it's, despite being important for protein synthesis, most dietary tryptophan goes down the kynurenine pathway. And it gets increased with inflammation. So normally it's going through with TDO, but the IDO enzyme gets activated with inflammation and pushes it down. So it stimulates that. So then you get low levels of tryptophan and high levels of kynurenine. So you have a high kynurenine to tryptophan ratio. So a high KTR ratio has been associated with a higher risk of diabetes, higher BMI, obesity, inflammation, AIDS, cancer, sorry, the pregnancy's on this, all-cause mortality from cardiovascular disease, and it's correlated with interferon gamma activity. And the KTR ratio has been called the gold standard of determining IDO activity. So it's something to look at. This also is a great article by Tan. It is actually on ALS, but they do a really good talk about the kynurenine tryptophan processes. So the ones that are in red are actually considered neurotoxic. The ones in green are neuroprotective. The ones in blue are both. So another thing about the tryptophan, it may that when you, an age-related cause. So there's inflammaging. So as you get older, there's more inflammation, and that can cause tryptophan to go down and kynurenine to come up. You have a decrease of NAD production and an increase of possible neurotoxic kynureninic metabolites. So looking at it another way, another great article, tryptophan availability, it comes in from the diet. It can be, used by the microbiota to make indole compounds that have different impacts, or so you want to have a healthy microbiome. Forgot I had things that come up. Or it can get absorbed. So you want to make sure you have good digestion and absorption. It can go to the kynurenine, or it can go over to serotonin. But if this is increased, this will decrease. Interestingly, lifestyle, like weight loss in studies, has resulted in a decrease in KDR along with a reduction of the other markers, but the ratio decreased, and then an increase in vitamin B6. They weren't necessarily eating more, the availability was increased. And then lastly, this is looking specifically at tryptophan and types of protein amino acid ratio. So this is the effects of proteins and carbohydrates on the influx of tryptophan, and this is specifically to the brain. So the green arrows are going to identify an activation of carbohydrate-induced pathway, and that's going to result in increased muscle uptake of large neutral amino acids, which in turn increases the plasma ratio of tryptophan to large neutral amino acid. So tryptophan, it's very small in quantity. So if you have a bunch of these other large neutral amino acids, it has a hard time getting across. And so you want to keep that level so it doesn't overwhelm these. So the red arrows are indicating things that are normal dietary protein sources, and they have a lower tryptophan percentage content. and that decreases the plasma to tryptophan large neutral amino acid ratio, so you end up getting less in the brain. So tryptophan has trouble competing. So just trying to look at how you can make sure to increase the tryptophan. And this is my last thing on tryptophan, which I find really interesting. So more about, the interferon gamma leading to IDO activity, which causes the kynurenine, the tryptophan, to go up to the kynurenine pathway, and reactive oxygen species. And what it can do is it can impact the BH4. So remember, phenylalanine and tyrosine use BH4, but tryptophan uses BH4 as a cofactor to go to serotonin. Arginine uses it to go to nitric oxide. So if this inflammation starts, it can slow these pathways and increase the other ones. And then looking at genetics type marker. So when you're looking at metabolomics, and this study was interesting, this is from 2018, and they looked at people who have ApoA2 gene, and it's a marker for obesity based on diet. So essentially, people who had the genotype TT or CC, if they were on a low saturated fat diet, they were fine. That's, nothing really got activated. But if they went on a high saturated fat diet, those with a TT genotype had an increased risk of obesity. So this is the person that I been working on and talking about, so our accountant. So looking at some markers that to me provide evidence that she is on a plant-based diet. So citric acid was very high, so that is associated with a plant-based diet. Her tartaric acid, which is higher, it's a marker, could be of wine intake. It is used by organizations as a marker of wine intake, but also grape juice. 1-methylhistidine is a marker of dietary meat and fish intake. 4-hydroxybenzoic acid is we went over that. It's a marker of just anthiazine markers, so purple type of foods. Hipurate also goes up with polyphenol intake. And then methylmalonic acid. So that goes up with B12 issues. So also you see issues with B12 when somebody's on a strict vegan diet or even more plant-based focus. And then she has some other areas of concern. Her tryptophan was low, her lysine was low, glycine, and so she had some moderate inflammation. So this is her, actually her GI map test. So it kind of is helpful too. So her lactobacillus was low, her Clostridia, which some of those conversions require some Clostridium. Her digestion's a little impaired, so she doesn't have any steatocrits, so fat's getting digested, but elastase is a proteolytic enzyme. And sometimes it's obvious, lower population studies, elastase is lower with people on vegan, vegetarian diets, but most, once you go below 200, there could be some pancreatic insufficiency. Immune markers are fine, but she may have a little bit of leaky gut, all things that could impact her picture. So I'm looking at her glycine. So her glycine level was low, and her threonine level is okay. So it's not that she's not getting it, so it could transfer over, and serine and glycine also interconvert. So, it's pretty low, so it must be getting used up. So I'm going to look at different things. It could be a glutathione issue. It could be a conjugation issue. So either with toxins or even, glycine can conjugate with benzoic acid. So if it's low, generally you're going to supplement. Also, because that B12 is there could be an impairment in methylation. I'm also going to look at these things that I wondered about. So methylation, glutathione, and it's nice to have these all together. So the top half is looking more at methylation markers, that involvement of the pathway. And then the bottom half is looking more at glutathione production. So we'll look at the top half. So elevations of cystathionine have been associated with certain vitamin deficiencies, B6, B12, and folate. And we already know her methylmalonic acid is elevated. So I'm going to put money that it's more likely to be B12. So it's also associated with other health conditions, which I don't think she has at this time, but these are things that I would look for. And then cysteine. It has been noted as a rate-limiting marker for glutathione production. So cysteine and sulfate together feed sulfocysteine, and sulfocysteine going up can actually deplete cysteine. So you'll see here that cysteine comes through, and then cysteine is going to feed it, but every time it comes in, glutamic acid or glutamate has to go out. So the other part that I'm looking at is glutathione. So glutathione is, an intracellular antioxidant. It's made up of glutamate, cysteine, and glycine. And so it can have an impact. So I'm looking at some markers. Cystathionine is metabolized to alpha ketobutyric acid and cysteine, and they can be markers of glutathione production or inefficiency. And then pyroglutamic acid is produced at the end steps, and it can be related to inadequate amounts of cysteine or glycine availability. And we know her glycine is low. The other thing that she had was the low lysine. So lysine catabolism leads to collagen and carnitine production. So it also assists in absorption of essential nutrients. So plasma lysine and glutamine levels and higher glutamic acid were also associated with ADHD rating, and she thought she might have some attention issue. The thing with glutaric acid, when it becomes elevated, and hers is within normal range, but when it becomes elevated, it gets detoxified through carnitine, so it can lead to some carnitine deficiencies. The other area that I'm looking at is her tryptophan. She has a good amount of 5-HIA, so that seems to be working, but her KT ratio is elevated, so she may have some increased inflammation. The rest of the pathway seems to be pretty good. So maybe some inflammation. I don't know if these styrene exposures can impact inflammation or if they are impacted with glycine conjugation. The other thing, this is an interesting study, and it looks at tart cherry juice for insomnia. And the reason that they think that it works for insomnia is that it's likely, this procyanidin B2 is likely an active ingredient in tart cherry, and it acts by reducing the kynurenine to tryptophan ratio. So as that ratio comes down, inflammation comes down. And then when you inhibit that, you're going to get more over to be able to go to serotonin pathway, which can go to melatonin, and less coming down that kynurenine. So reduced inflammation may be partially responsible for improvement in insomnia. So I'm going to review her diet. I'm going to make sure she has increases in antioxidants. I'm going to look at the cherry juice. I'm also going to look at cruciferous vegetables because she had a specific SNP that would benefit from cruciferous vegetables when I did her steps. She had a GSTM1, so it's normally going to detoxify chemicals in the body, and studies have suggested that people with that genotype would benefit from eating more servings of cruciferous vegetables. So I'm going to have her increase those cruciferous vegetables. And it's easier to get people to stick with things if I can show them reasons that they should do that. So additionally, I'm going to consider a multivitamin with the B12. I'm going to consider glycine, lipoic acid, and acetylcysteine, and some glutamine. Going back to her gut issue, I'm going to get her a lactobacillus probiotic and digestive enzyme. I'm also going to try to track down where those styrene containers probably to go. It is pandemic. And then I'm going to monitor those metabolic changes. So this is going to just be part of the what she gets as normal health every year. So you can see if there are changes and see what's different. So that is that. So now I think I'm ready if there's any questions.
Excellent. Thank you, Dr. Redmond, for a great presentation today. And our first question today is, are many of the metabolites produced by microbes in fermented foods the same as beneficial metabolites our body produces? And if so, how significant are fermented food metabolites to our diet?
Yeah, I would say that they, I don't know if they're the exact same, but they're certainly going to have an impact. Last summer, I know the journal Cell had a big study on fermented foods, and they're like, everybody talks about fiber, but don't forget fermented foods. So I didn't look specifically on what impacts that would make on metabolomic markers, but I would think that it would have that. So they're making it in fermented foods or our guts. So I would assume it certainly is going to have an impact.
Thank you. And someone is asking, who would benefit from this test?
Oh, every everybody. Um, so it's not the type of test like people who have, well, certainly people who have metabolic disease. So there's a lot of research on metabolic, cardiovascular, and those type of things. But I have seen in some of those research studies that maybe people should just start getting this as part of their yearly protocol, um, their yearly physical. So you can see how do you respond. So when you go take a test, if something's out of whack or elevated or low, if it's if it's you won't know is that normal for me or not. So when you're in good health, you can take it and have yourself as a comparison. So I would say anybody. I mean, I would really encourage it for people who have weight loss resistance or who are worried about risks of metabolic type diseases, or there's a lot of research on cancer too. I have a hard time trying to pinpoint one, but I think it can be helpful for everyone.
Thank you. And is there treatment recommendation that comes with this test report?
There's a summary on the back that says, what nutrients may impact it, and it looks at specific areas like kidney impacts or inflammation. And so we're developing more literature as we go. So, we'd love to know what's helpful. We have a kind of a cheat sheet. It's a 16-page cheat sheet on what some of the markers mean when they're high or low. And then we're, hopefully soon coming out with some more.
Thank you. And someone is wondering, what metabolomics test did you use in this presentation?
Oh, I used Diagnostic Solutions or Organics Metabolomics. So it's a new test that they have. And it has, urine, organic acids, and plasma amino acids. It also, you can get the amino acids in blood. So they have, 4 different versions of it. So it is available.
Thank you. And how quickly would you see changes in the test results with dietary and lifestyle changes?
So, in one of those studies, they saw, a decrease when they really worked at it in 7 days, but I wouldn't have somebody make changes and do it in 7 days. So, I usually think if you're going to do the test and make a lot of changes, I would do a follow-up in like 3 months. We've always said like 3 to 6, just depending. But yeah, it's kind of like A1C. You can see a change in A1C pretty soon, but you can't want to see a big change and a real steady change after about 3 months. So I would say about 3 months.
And someone is wondering, how are urine only versus plasma and urine collections for the test different?
So the difference, you'll get your amino acids and a few other markers in urine instead of plasma. So, years ago, urine was harder to get information on, and it is impacted by diet. So either one of them, you want to keep your lifestyle the same. And a lot of them do correlate, some of them don't correlate as well. So it used to be that the research was primarily going to be on plasma. So there's more research on urine. So, I've talked to people who do urine tests. You can get good information out of it. I prefer the plasma just because it does have more research to fall back on, but it's not that getting the urine doesn't give you good information.
Thank you. And someone is wondering, what are the dietary restrictions for sample collections?
So we prefer fasting samples. So for the urine, it's first morning, and then it's best to have a fasted plasma sample.
Excellent, thank you.
Mhm.
And it does look like that is all the time that we have for today's program. And thank you again to Dr. Redmond for a great presentation today. And thank you to all of our attendees for taking the time out of your busy days to join us.